Hello guys, today topic of discussion is the cell cycle regulator, right? So before I'm telling you about what are the cell cycle regulator, I want you to explain about the apoptosis and how cell death is occurring by the process of apoptosis. So what is actually apoptosis? Apoptosis is the ATP dependent program cell death, right? ATP dependent program cell death of the cell, right? So how does it happen? It happens by two ways. One way, which is called the intrinsic, one is intrinsic pathway, which this picture is showing over here, and the other one is the extrinsic pathway, like extrinsic pathway. So now tell me about this apoptosis process, starting with the intrinsic pathway. So what is happening? Normally, this pathway, this side of the picture, it is showing normally what does happen. And when the cell is damaged, what has happened and how the cell is going into the apoptosis, right? Over here, normally what is happening to the cell? Let's say this is viable cells. This is not undergoing apoptosis. So what is happening? Growth factor, they get attached to the growth factor receptor, which is present on the cell membrane, right? This is your cell membrane. So when growth factor attached to the growth factor receptor, it causes growth of these cells, right? It wants that cell to survive, right? So what does normally happen when the cell need to be survived? There are two proteins which need to be checked. These are called pro-apoptotic and anti-apoptotic. They need to be checked, right? How they need to be checked? Like pro-apoptotic means they are causing the apoptosis of the cells, right? And there is anti-apoptotic genes which inhibit the apoptosis of the cells. So this is quite understandable that when growth factor is attached to its receptor, it causes these proteins to be activated, right? Anti-apoptotic, because the receptor or the signal is coming that wants cells to be survived, right? So what these proteins, the green one over here, what they does do, they will keep binding with the pro-apoptotic, these protein, red one, which is backs and back, right? They keep the green one, which is anti-apoptotic, they bind with the backs and back, and they don't let it activated. So when they are activated, they open the gate. So they don't let these red proteins to be get activated. So when they are not activated, they are present all on the mitochondria. Okay, this structure is your mitochondria. So anti-apoptotic BCL2 green one, they bind with the apoptotic protein, which is backs and back, on the cell surface of the mitochondria. So what is happening? They are basically, they keep the cytochrome C. This cytochrome C, the blue dots you're seeing over here, they keep the cytochrome inside the mitochondria. They don't let it come outside, right? Who don't let them come outside? Your anti-apoptotic protein. How they don't let it come outside? Because they keep these backs and back proteins binding with them, right? When they're binding with them, they don't form a gate, and through that gate, the cytochrome C is normally released, right? So growth factor bind with the receptor, it causes the cells to be survived. So BCL2 bind with the backs and don't let the cytochrome C released, right? So cytochrome C is not released, that gives the signal that the cell should be survived. That is what that is one thing happened. Now how can you remember that how backs is causing apoptosis and BCL2 is causing anti-apoptosis? So I make mnemonic. I told you guys that BCL, this is C for cell, right? And L for live. So this is my way. I remember this one, that CL means cell live. So BCL2 and BCLX, they want cell to be live long, right? Cell live. And backs and back. So backs is basically having X, right? So X means you are targeting something, right? You are targeting something. So this way you can remember that this is pro-apoptotic. One thing. Now the other pathway, now the coming toward the apoptosis, how apoptosis is happening. So you can see over this picture, when the cell is damaged, that is one point. So when cell is damaged and you don't have the signal, what is the signal? This growth factor, right? You don't have any growth factor over here. So what is happening if your cell is damaged and you don't have or you lack of the survival signals? These both things causes activation of this protein, which is called BH3, right? This protein is activated. Now, in case of apoptosis, what this gene, when this is activated, this lead to the apoptosis, right? Yeah, now you have to understand how this apoptosis is occurring. This protein do two jobs. Once it bind with the BCL2 and BCLX, right? As I told you, cell life. So these are the anti-apoptotic proteins. So it binds with this one and make it inactivated, right? And other thing, it binds with the BX and BAC and causes activation of those proteins, right? You see over here, they bind with the BCL and causes inactivation, binds with the BACs, causes activation. So when these proteins get activated, they open their channels. When these channels are open, what is happening? The cytochrome C is released. Cytochrome C is released, causes caspase activation, and apoptosis is initiations, and then you have nuclear fragmentation and babbling, and all that will lead to the cell damage. The other pathway, this is the intrinsic pathway. This side is the intrinsic pathway of apoptosis. In extrinsic pathway of apoptosis, I'm just mentioning over here. So you have fast receptor, fast ligand. It comes with the fast receptor, and what it causes? It causes the activation when the cell is damaged, right? When the cell is damaged, fast ligand comes at bind with the fast receptor, causes activation of the initiator caspases, which causes activation of these other caspases, and that release the cytochrome C, and all that process is the same. The difference is which why when ligand is bound with the receptor, it causes the activation of the caspases, and then caspases causes all that system, and they causes the release of the cytochrome, and which lead to the babbling and the cell death. That is the apoptosis. Now coming to the this cycle, that is our main topic, that is cell cycle regulator, like how body decide that this cell should be survive or this cell should be uh, not undergo the cycle, right? So this is your cycle. What is that cycle? This cycle showing you that when cell need to be divided, right, as your skin cells, so skin cell, hematopoietic stem cells, your neurons, or other cells, right? So what does happen from G naught, cells enter to G1, G1 S phase, S1, G2, and mitosis, and the cells is divided and producing the two daughter strand, right? Under two daughter cells. So this is the cell uh, growth, right? Cell growth. Now there are certain regulators which check this cycle. These are the checkpoints, right? These are the checkpoints. So what are the checkpoints in the cell growth or the cell cycle? So they are called, there are cyclin dependent kinases, right? These two are, and the third one, there are P53 and retinoblastoma. P53 and retinoblastoma, these are tumor suppressor gene, and also cyclin and cyclin dependent kinases, they also check. So what cyclin dependent and cyclin dependent kinases they do, they basically tell that this they are activated or inactivated depending upon the situation, that this cell should be undergo progression or this cell should be undergo apoptosis, and their check is balanced by the P53 and retinoblastoma. Let me explain you how they are doing this thing, right? Okay, so what is happening in this case? So what is happening? Let me wipe it off so you understand. So you have to understand the normal, what is normally happening. So in normally, so cyclin, it is present inside the cell, so it is normally inactivated. When cyclin bind with the CDK kinases, CDK kinases normally when they are found inside the cell, they are inactive. But cyclin, when bind with the cyclin dependent kinases, CDK cyclin dependent kinases, they causes activation of this CDK. So CDK is doing what? It causes phosphorylation of the retinoblastoma. So this one, pink one, is your retinoblastoma gene. So normally retinoblastoma is attached to E2F. So E2F is what? It is your transcription factor, which is called elongation 2 factor, right? Retinoblastoma bind with elongation 2 factor. So what happens when cyclin bind with cyclin decay? These are activated. When these are activated, they do what? They causes this retinoblastoma attached with the phosphate, that is causing hyperphosphorylation of the retinoblastoma. So when they causes hyperphosphorylation, that this retinoblastoma become inactive, right? So this E2F is released. When this E2F is released, because now phosphate is attaching the phosphate, E2F is released, E2F go to the transcription to the DNA, and it causes the gene transcription, and this is how the cell is going into this phase, like causing the cell growth or the division. So that is normally happening. So confusion point over here which I want to explain you guys is that this part, when you say like how this confuse it, like cyclin when combined with cyclin dependent kinases, it causes what? This E2F retinoblastoma to bind with phosphorus, and this arrow is showing, like this arrow showing that the retinoblastoma is going to attach with the phosphorus and become inactive, and this E2F is now released from this retinoblastoma and attached to the gene and causes the transcription of gene and the cell cycle growth, right? So what is happening? Let's say this cell, it is undergoing cell, their DNA is damaged, right? So when DNA is damaged, the body don't want that cell to be survive or undergo the multiple mitosis and produce more progeny, because this is basically damaged DNA. If it is not repaired, that cell should be stop growth and it should be undergo apoptosis, right? So that process is maintained by this. Uh, process is maintained by your this P53. Now what is happening? Let's say the cell is damaged. This is the signal the cell is damaged. It causes activation of P53, right? So P53 does two jobs. It causes induction of this P21. So P21, what it does? It combined with this complex, right? Which is normally causes retinoblastoma inhibition by phosphorylation and leaving of a E2F factor, right? So it P21, it binds with this complex. So when this complex is bind with this one, what is happening? Retinoblastoma is now releasing, which is initially inactive. It release the phosphate. Now it go back and attach with the E2F, right? When it go attach with the E2F, now E2F is not available on the this side on the DNA to cause cell progression. So that is how cell cycle growth is stopped. P21 bind the cyclin and CDK and causes inhibition of this complex. Then retinoblastoma is going hypophosphorylation or leave the phosphate and get attached with the E2F, and E2F is now attached with the retinoblastoma, and this progression will be stopped. One job. P53, which is tumor suppressor gene, it does the other job. What is do? Now the cells, this cell is stopped, right? This cell's growth is arrested. Now body also need these cells to go undergo the apoptosis. Now the cell is stopped. So this is also the job done by the P53. How it does? It causes inhibition of this BCL2, BCLX. That is also as I call cell life. So cell life proteins are inhibited by P53, mean they are causing now this cells. It doesn't need to be survived. It undergo apoptosis. So what is happening? Normally BCL and BCLX, they inhibit this apoptotic protein. When they are inhibited, this check is gone. This check is gone. So what would be happening? They are activated. When this apoptotic protein are activated, they causes caspase activation, release of cytochrome C, apoptosis, and that is how cell death is occurring. Hope so this lecture is helping you guys. Thank you so much.